from EPSCI. Tycho didn't know what EPSCI was, so here's a picture of it. It's a famous school in Paris. Marie Curie, I think, did, Marie and Pierre did some of their early research here. The gentleman was director for a while. You know, lots of famous people who were there. And now it's Jerome.
Yeah,
all these labs over
yeah,
here.
by chance it was a picture at the right place.
Jerome was a graduate student, got his degree from University of Bordeaux, wine country. He was a postdoc at Exxon with Dave Waits when Dave Pine and I were there, and he already had a reputation in those days. He then went back and he was a professor at Bordeaux, and in about 15 years ago, Dijon seduced him away from Bordeaux to come to Paris and work at EPSCI. He's known for lots of things, especially emulsions, fantastic work in soft matter, but also as an entrepreneur. His company, which he recently sold, one of his companies, which he recently sold, is Capsum. You go to their website and you get these credos here and stuff like that. You make amazing materials using soft matter and stuff like that. The main thing that really got me about this was using techniques like this emulsion formation here. He made these puffs, these artificial caviar, if you like, and this is really amazing. Now I'll tell you what's really amazing about this. This is hand cream, $1 for a liter, right? But you encapsulate them in these little gold balls, okay? And it's essentially $500 an ounce. So he's a real genius in terms of not only science, but as an entrepreneur. And he sold this company to
Chanel, a few weeks ago, by the way.
What do you do with these things?
It's hand cream. You take it out, it turns out you can buy it with a silver spoon. You take this out, you put it on your hand, you rub like this, it breaks the emulsion, it breaks the droplets, okay? And now it's hand cream.
I must have brought some samples.
The amount of value we added is a factor of 10 to the 6 or something like that. Anyway, okay. So now we'll let him give his talk.
Okay, so thank you.
Science, not just hand cream.
Reconnected?
You should be, you are. Yeah, it's up there.
Okay, good. So thank you very much. Of course, it's my very big pleasure to be around you, and the story has been very long. I was there about 9 years ago for a couple of months. We were remembering that with Paul a few minutes ago. And this place is very familiar, to be honest. The city is familiar as much as Paris. So some years ago, I was knowing more New York than I did in Paris. Okay, so Paul, when he invited me, it was a real excitement, and I tried to put things together such it will be entertaining. And please, if there's anything which is not very clear, you interrupt me. It's mostly movies, stories. There is not so much effort that I'm going to ask you. Just listen and watch the movies. Okay, so the title is the one that Paul came up with, beautiful one, Physics and Fluidics applied to metabolism, cell population dynamics. So what we are going to explore is how we can use physics, fluidics, soft matter science to bring new technology, new ideas, to learn about biology. I'm just a technologist, we say in English, yeah? Trying to bring something in biology. I'll never be a biologist, so I can help thinking about ways to help biologists to do discoveries. And if I'm part of it, I'm very glad. So we start to build a group in Paris with real biologists joining us now for good. Like people like Griffiths a couple of years ago, he was in England, and a recent one called Paul Rene, he was Oxford and New Zealand. So now we start to have the real person to share our ideas. Okay, so I have 3 stories to tell you. It takes a bit less than an hour. And the first one is concerning the adaptation and what is called very pedantically adaptive radiation in a microbial population under stress. So what does it mean? It means that microbes, when they are under stress, they are evolving. And in a very complex manner, the number of microbes could be as big as 10 to the 10. So I propose, as a colloidal scientist at the beginning, to try to put light on that, the same way as if someone would ask you to tell what's going on for each of the colloids within a batch in time. It turns out the bacteria is a bit more complicated than lattices, but the idea is basically the same. Okay, my friends are all listed here. I just want to mention these two brilliant students, and Denis Cotinet was really the champion of this work. And you have a list of postdoc and my colleague in Paris, these two young scientists and also the biologists joining. And Arjan was been bringing all the strain of bacteria. Okay, I like to start like this. At the end, as a scientist, you want to answer questions. And there is one thing we want to answer is how the nature did evolve and why it's evolving so fast, why the species are so numerous, and why when there is a big accident, it's reflourishing new species so fast that it's above our imagination. So I propose as scientists to say, what don't we set up technologies to try to put some light on that? And at the end, what we want to explain is how species evolve, maybe not the most complicated species. So I'm just putting as a sort of funny game the idea of Lamarck. He was an ancient naturalist, and he said that driven by inner need. So the idea was because the giraffe wanted to eat in high trees, it took time, but the neck get longer. Okay, it's not so stupid at the end. Because, you know, the organism is very complex. The environment around this animal is very complex, and at the end, nobody can tell today how this is evolving. Then a couple of years after, another guy, which is very, very known too, is Darwin, and he came up with the idea, based on this observation with the finches of these birds, that in fact it's called all work with the natural selection and all the Darwin theory. But as you know, to get the Darwin theory applicable, you need to have reproduction, growth, and heredity. This is the three criteria that are strictly necessary to apply the Darwin evolution theory, which is not always the case. So it's opening so many questions that I would say that it's far from being solved.
Growth of the population.
Growth of the organism.
Oh, not of the
He has to grow, okay? He has to keep the heredity of the parents, and he has to divide.
Bacteria don't grow.
They do. Bacteria, they divide. They divide, they grow a bit, redivide, grow a bit, redivide. There is an exponential growth of the bacteria when it divides.
That's just part of its reproduction. Right.
There is reproduction, growth, heredity. In bacteria, it's fine. But for many other species, it is very difficult to distinguish between growth and reproduction. So, you know, all that seems clear to everyone, but it's far from being clear. That's why I think that bringing our brain into it is not a loss of time. Okay, so anyway, we have at least one possibility, which is using microorganisms to try to maybe understand a bit better. But one thing is sure about microorganisms, there is a very large probability that Darwin was right, because we have the three criteria and the environment is much more simple. So it's I agree that it is far right for microorganism communities. Okay, it's interesting to play with microorganisms because they grow fast. You know, the dividing time could be as small as 30 minutes for bacteria. So we are expanding a lot the possibility to observe what's going on with experiments that we can run at our scale. But the problem with microorganisms is that we don't know what is a phenotype. That's mainly the question which is addressed. I don't list everything on my slide because I think that everybody knows dividing time, the yield, and so on. So here is some slide which is important to follow. You know, if you look at microorganism, you have a bunch of chemicals into this bag, which is a bacteria. Don't forget that the bacteria is just a membrane. Everything is mixed up inside. There is no compartmentalization. There is 1,000 of different proteins. It's all work together, just by affinity and fluctuation. So out of this composition, then you have all the interaction between these two components. And then at the final, you have what you call the phenotype. But how do you define a phenotype for bacteria? For evolved animal, it's easy. It has some characteristics, the eyes or whatever. But for bacteria, that's a very big question. And before answering that, or having some guess for that, it's hard to go into the field. Okay, so one thing that has been done for many, many years since past time, it's plating. So what's about plating? Plating is putting single cells on a gel. You wash things out, so you have only cells very well separated. And you wait for growth, making kids and kids and kids. And at the end, you have shape of colony which might be different. And that was raised very nicely by Paul René, which has joined us now. And he made some type of bacteria under stress, and he showed that they are evolving to different species recognizable from their shape on agar. Okay, this is only phenotype so far we know for bacteria.
By phenotype, you really mean something that distinguishes one bacterium or one group of bacteria from another.
Species.
That's what you mean.
Yeah, but you know it's vague because they don't cross-reproduce, so we cannot make a distinction from sexual reproduction. So it's a species difficult. It's just wording. We have to almost define our own words in order to go into the field. So
when you say evolving, you could have a certain population, and then you could have selection, and then the ratio of the one that has the advantage.
We'll keep going.
But that doesn't include, evolution includes more than just that. So when you say evolving, I don't know exactly what you mean.
Evolving means that there is a stress, and any change in the composition, in my previous slide, genome or whatever, will give some advantage, and if there is advantage, it will take over the other one, and then you are converging to a new species. That's what evolution is. So the Darwinian language is very simple and useful for us today. So okay, so you see this phenotyping has been brilliantly demonstrated by Paul, and he showed the different species, and this is basically what all the pathologists are doing with bacteria. They play them to see if the colony resembles the ones they know. If they don't resemble, they are just get afraid and go to some treatment. Okay, so we want to overtake this limiting step. This is basically what we want to do first. I make it very short because I'm not a biologist, so I understand things as a physicist very from the top.
Could you try to talk a little louder consistently?
Yeah.
Your voice drops out.
I'm sorry. I try. So here is what the postulate is for us today. We say we have a very complicated bag full of different molecules. Okay, this is defining at the end. If you know everything into this bag, you say, oh, this is a phenotype. Okay. But what we want is to read the phenotype from outside. And we postulate that all this complicated network of genes are interacting together. If we put a reporter somewhere on the genome, if something is changing here by evolution, because something has changed in this complex system, it will affect also this reporter gene. Postulate. Because everything is coupled together, and if there is severe change due to evolution, we should be able to read it in time directly into the system.
This is a picture of a single bacteria.
This is all the coupled network of genes in a bacteria. One.
This also.
And I simplify this stuff by this, right. And I say I add one gene which is emitting this fluorescent protein, and I postulate that if everything is changing here, it should change that.
Right, but it's not a population or
this is a scheme of the coupled network of genes in a bacteria. A scheme. There are probably some stuff to discuss about that too. Okay, so let's take coli, Escherichia coli, a bacteria.
So which kind of gene is YFP?
YFP, it's a gene that codes for fluorescent protein. Okay, so it's inserted into the genome or in a plasmid, whatever. If it is there, it will produce some amount of the fluorescent material. And this will be very easy to read. And if this amount changes with the species, then we are winning. Okay, so we have this Colli strain provided by Arjan, and excitation, emission, and so on. Okay, I go next. But the problem is the following. If you take this bacteria population, if you send it to a cytometer, everybody knows what is a cytometer? Okay, it's an instrument which is 30 years, 40 years old, which is used every day to make your hematology mapping, take your blood, and I will read every single cell and the characteristic of all your blood cells and give a map. That's cytometer, hematology, whatever.
What do you measure about the cells?
You measure the size, the granulosity of the cell by looking at the light which is scattered in different directions. This is hematology, or you can even measure impedance of cell going through a very small canal with nicely located electrodes and so on. So if you take this modern cytometer, I take the bacteria and I observe them one by one, and I plot the distribution of the emitted light due to the transcription of this protein, and what I found is a distribution, but the CV is at least 50%, so there is a big noise, which is called the biological noise. So the problem is, if we define the phenotype as this emission, we will be screwed up by the biological noise, which is there because there is fluctuation into the cell, the amount of polymerization is changing, and so on. This is very known, well documented. Okay, so we have to overcome this if we want to do a job. So this is just to keep you awake and make you understanding that this biological noise make impossible to measure a phenotype instantaneously. Of course, if you grab a cell and you average over a long time, suppose that the cell will be maintained the same stage, you could do it in principle, but this is typically impossible. However, as for plating approaches, when you put a single cell on a gel plate, if you wait for the family to be built, then you read an integrated property of the ancestor, which reveals the phenotype of the ancestor. Everybody understands that it's critical.
So your assumption is that nothing changes the progeny identical to the ancestor?
Any inherited traits can help. If it's inherited, you can amplify.
But the genes are still regulated by the environment, so maybe you get different expression if it's in the middle than the edge.
In a colony?
Yeah.
Yes, in addition. That I hide that. In addition. But the principle is maintained here. Okay, next. We have to develop a technology, and I go very fast on the technology because it's not the purpose of my talk today. So the technology is basically creating a train of drops. So this has taken almost 10 years to develop, so you can ask any question you want. Basically, what we develop is a technology where you have reservoir of the nutrient, a spacer in between. This could be gas and a type of oil which is not miscible in other oil or whatever. The quality of this instrument is basically absolutely no contamination from one reservoir to the other. Why? Because it's lubricated, it's a good wetting property, and it's continuously flowing, so everything is maintained properly. And at the end, it works very well. It's a sort of instrument to analyze cell one by one, just on a one-dimensional tube, which could be as long as 20 meters on a coil and so on. So if you want one more or two more information, this is the machine, how it works, just to give you that is easier than talking. So you have coils that can go over 20 meters of tube here, and the train is continuously flowing from one coil to the other and passing through the detector. You grab the information over each reservoir and you stack that into your computer. And you can have as many readouts you want. Okay. First results. If you start with single cell into these drops, single cell, so you play with the Poisson distribution, so most of the drops are empty, we don't care. Now you keep only the one which was filled with one cell. If you look at the growth curve of each single cell within this drop, which is 100 nanoliter in volume, you discover that if you start with single cell out of the same clone, you see that even though they are totally separated, they grow exactly the same. So that was, for me, quite a surprise. This complex bag of molecule starts same moment, burn the same amount, and make the same amount of fluorescence of your reporter, which is a YFP protein. If you plot here the distribution of this yield here, you see that it's almost a peak, and the resolution is basically the one of the instrument. Okay. I remind you that before we had a large potato when you see the cell one by one. But now if you take the cell, you let the family grow, you integrate everything on the family, you get this, you get this. Okay, just to confirm, this is what you get from putting the cell one by one into a cytometer. You go into this device, I show you the millifluidic device, you get back to the instrumentation limit. So we can go back down to 1% if we make some progress. Amplification and near the biological noise. Okay, so that makes us very happy, because now you can start to believe that you can phenotype a very complex system. Okay, so now I'm going to tell you in a very rapid way one experiment we did by forcing the system to be under stress. I skip this. And we revisit an experiment which is a very ancient experiment, which is taking a bacteria population, let it grow to the stationary phase, close up, and let it age. No more food, no more anything. So the bacteria, to survive, have to eat the cadavers. That's how it works. And there is an experiment which has been done. If you enclose the bacterial system, 5 years after, there is still a large amount of bacteria still alive, because they recycle everything. But to recycle everything, they have to evolve, because the food is always changing. And the niches are changing continuously, so they have to find solutions to keep them alive. And that's what we are going to probe with our experiment. So you start with clones, and you make different tubes. You open the tube at different duration. Every tube is independent, because you have to shake it up to sample. So you can take the same tube for a different time, okay? So each is another experiment, another world that you are going to examine at the end. Okay, so if you take your cytometer again, from 1 to 30 days of starvation, you look at the expression of the fluorescence protein with the cytometer, nothing changed. So you would say, okay, there is no aging.
What are these different? These are
different
days.
Yeah, this is 6 days, 15 days, 30 days, and it doesn't change, so it doesn't make real progress. But now you do the same experiment, and you put in the millifluidic device, and that's what you get. As a function of time, you see that the cells that you are taking from the system, putting into the millifluidic device, let the family build into the good food, and you find that they express the memory of the parents, and you have different classes. So what does it mean? Classes, species, we reveal classes with a readout, but if these classes are there, it's because they have advantages. Here, this is time. So this is a time, the measurement in the millifluidic device is done over a few hours.
Okay, I just couldn't read the unit.
So yeah, I agree. So I'm sorry. So this is a few hours, and this is fluorescence intensity in unit arbitrary after renormalization. What you discover, you...
Can I compare the arbitrary units between axes, though?
Yeah. It's all renormalized by the same amount.
So if I look at the day 30 ones, they grew to a larger population than the day 1 ones?
Sorry?
If I look at day 30, did they grow to a larger final population than day 1?
So day 1 is here.
Right. So
at day 1, I pick them, I put it in the machine.
Right.
That's how the cell grows in a few hours.
Right. So I'm just saying it saturates at arbitrary unit 1.
No, it saturates here because we decide this will be 1 in our units.
Okay.
And everybody is renormalized compared to this. And this is the amount of fluorescence you have a saturation when the food is exhausted into the drop.
Right.
Okay. Everybody follows.
No, I'm sorry.
Two.
It's 2. It's
What? 2 what?
In, the y-axis is 2. Right?
The ordinate.
Yeah, so there is more fluorescence. Be patient.
Yeah, okay, so that was...
More fluorescence. The gene is producing more because coupled to the network, there is some good reason that making more active.
Sorry, I didn't understand what I'm confused about what the experiment is exactly. So you start off with a system.
You have a large...
You're evolving all along, and then you look after day 1, or after day 3, or after day 6, and then of those ones, you start something new?
At a day 1 to 30 days, you take your sample, microscopic sample, shake it up, open up the bottle, take a bit, put into the millifluidic device, 1 cell per drop, analyze. And you have about 1,000 cells in each experiment you analyze. Okay? If you...
How many cells there are in drops?
In a drop, you start with 1.
The drop is 1.
In each drop, you have 1 cell.
Just 1 cell. Just 1
And it grows up to 100,000. That's how we make the measurement. I should have I've said it. So that's how we amplify.
So how do you know that it's grown until now 100,000? You measure in the D?
Because, yeah, OD you can, so that's easy. Typically, it's 1 to 100,000. This is the amount of food we put that enable to produce this amount of cells. It's clear for everyone? Yes. Because we are coming to the conclusion. You will see how the world could explode into diversity in a very crude experiment, but I'm almost sure it gives us a real direction to go further. Okay, so this is one experiment out of 15 different that we did, and each experiment we always found classes with some level of fluorescence, but the number of classes, how they are populated, the level of fluorescence is always different. Always different, except at the very early beginning of the aging, and I'll come to this. Now I put the distribution. Before we had just potatoes, now you have nice resolved peak just because we kill the noise by amplifying through the family in a good even food into the drops. Fine, okay, so now what we can do? We can do the following. The instrument has been developed such you can keep out, take out, sort the drop you want. The computer tells you, okay, this drop is in 1 class. I want to know something about this class now because we are playing with phenotype. And this is important. We are playing with species. As a gardener which is collecting the nice vegetable in his garden, we are not playing with some mental space. And for instance, it could be the genome space. And I want to make this parenthesis. So far, there is enormous progress on deep sequencing. So what the scientists are doing after a certain experience, this one for instance, they take all the sample, put everything in a prep, take the DNA and full sequence the DNA, and they will tell you, oh, we have all this bunch of mutation with this frequency and so on and so on. But where are the species? Disappeared. Okay? We keep the species alive. It's a very big difference in terms of scientific route. So now we can, after grabbing the species out of the complexity, we can say, okay, this drop, we can tell, what does it give if you plate it? What does it give if you go to the sequencer? And you can do all things you want, but the species is still there, and you can keep running the evolution of the species if you want. Okay? That's a very big difference. And this is answering most of the questions. So I take just one example. At day 30, we have these 2 classes. We can sort the drop. And if we sort the drop, send back to the cytometer, and we find once it's separated through our instrument, we are able to read a difference in the mean value of fluorescence with the cytometer. But if you don't make the sorting before, through the amplification and all this business, you find the potatoes that is impossible to resolve. And here it's an answer to the question I had. We can make, after sorting, optical density measurement, and we find that basically the biomass yield is the same. It's a really, in this experiment, a difference brought by the expression of this protein, which is linked to the network of genes. So we are coming to almost the conclusion of the first story. If you play it, you find that there are difference also. Most of the time there is difference. You see, this colony gives rise to bigger stuff than this one. But if you have all of that mixed, impossible to make any distinction. And you can say, oh, this one is bigger. Why? Because maybe it has a dividing time a bit smaller and so on. But whatever. And if you go to sequencer, another type of plating of this class, we discover that this is a mucoid-like phenotype that produce a lot of exopolysaccharide, which has some interest, by the way, for cosmetic market. And this one doesn't produce it, so it has a different shape. Could be distinguishable, this, with a good method, an image analysis. All right, and now if you go to sequencer, so don't look at the table, it doesn't make any sense. What you discover is indeed you have mutation. And on that, by far, we follow the Darwinian scheme, so there is always something happening in bacteria if you want to see change. So epigenetic is very limited. There is, of course, but much more limited. Basically, mutation, only a single base pair mutation is changing the phenotype. It's absolutely fabulous. The phenotype 1, the mucoid day 6 that we had before. Okay, let me go back here just a second for you. Day 6, you see, this phenotype is mucoid also. It's creating a colony which is like this one or this one. Okay, and if you go to sequencing, what you find is that in all the drop we collect on the same class, we find always one single base pair mutation in one single identical gene. That's it. This is able to cause the change toward a mucoidy, which was not known in coli, but it's what it is. So the mucoid at day 15 is still having the same type of phenotype, but in fact, it has different mutation. It's not the same gene, and it has two genes which has been attacked by mutation, and it gives rise to a phenotype which is almost similar. And besides, the biochemistry is more or less done. These genes are related to some mucoidy for some reason, etc., etc. In fact, there is a real strong connection between the genome space to the phenotypic space, but several different mutations in totally different places of the genome could converge to the same species. That's probably the most important I'm bringing here. Last slide, it's this one that I like. Out of this many experiments that we also repeated in conditions where we are steering the system during aging, not just relax, but we steer, homogenize. And then here we report every drop level of fluorescence we had at the end with a dot. And we invented this color code to tell the frequency. So what you see, every fluorescence level has been renormalized. So you see as a function of the aging time, the diversification of the trajectory of evolution you can find in a such simple experiment. So what you discover is the fact that the beginning is always a bit deterministic, and then you can go to a very large spread level of fluorescence which reflect all the possible pathway of metabolism the system is able to find. And why is it so different? And why each experiment is different? Because each experiment is an evolution experiment and depends on what has taken place early on. And it's go back to the very initial condition which somewhere give you the trajectory you are going to follow, and none of the trajectory are the same in different tubes. That's it for the story at the moment. So we can make a bit of computation on this and extract some function, but it's not the time to do it. The last comment I like to tell is about static steer. You see that in steer conditions, the diversification is much less rapid, much less intense, and it's normal because when it's not steered, static, you always have niches, and these niches multiply the possibility of coexistence of different species. So when you look at that, you say, oh, if in my mind, I make just a little effort to increase the complexity of these things and increase the complexity of the environment, you see absolutely the giant space to be explored by nature. And it's just a little story of what it is at the end, because we are dealing with bacteria, which are nothing in terms of complexity as compared to what we are. There is no epigenetic at the end. Okay, I'm done for the first story. If there is any question?
Sorry, so do I think about this static versus stirring case kind of like I would for macroscopic organisms, where like spatial separation allows for variance between?
Yeah, I think so. If you look at, for instance, the finch's story of Darwin, suppose the birds were forced to live all together and mixing up all the food and the resources, they will never come up with a diversification result. There is a story in the Lake Victoria also where fishes has been irradiating species like mud in about something like 1,000 years or less. And because they were finding niches, all different, so the fishes diversify. But if you mix it up, no, of course.
I have a hard time imagining that your growth media is that heterogeneous.
That the growth media, it's becoming heterogeneous if you don't mix. Just because gravity is causing change. And for instance, downstairs, the ecosystem will be different than upstairs.
Okay.
And upstairs there will be, maybe they have to stack on the surface, but there will be more freedom and food, maybe, you see? Okay, so thank you for listening. I'm switching to another story. Paul, what time is it, please?
You have about 15 minutes.
Left, that's it? Oh my God. Okay. So I just want to end this with this conclusion, insisting that mapping phenotypic diversity over time with the ability to sort species. This is really the most important I want to emphasize. And this allows to cycle species having the right phenotype. We can take them out and we feed some machine like that and go to an evolution strategy. So more to come. Paul will see the evolution machine in Paris soon. Okay, now I will tell you something about a totally different but very entertaining also, how we can learn about phenotypic diversity, this time concerning eukaryotic cell, our cells, and learning how they differ one to each other in creating tissues. So I'm just going very fast on that, but we are trying to opening routes to make this feasible in terms of studies. Okay, so thank you a lot to this nice student, my collaborator, and the Curie group who has been participating a lot into this. I skip this out because it just tells you that today if you want to recreate tissue out of this eukaryotic cell, you have to play with very subtle things, and for instance, the only technology which is available today is called hanging drop. So it's basically a plate, a microwave plate, where there is teeny holes, and there is by capillary force, there is a liquid which stick on the wall, and the tissue, by some miracle, like to grow in the meniscus. And there they fit very well, you know. You need a nice sensing of the environment to make these cells happy, so that all is available today. So we revisit this game by saying, why don't we invent a way to make sacks, bags, or whatever, made out of very thin micro hydrogel, totally biocompatible, totally permeable to anything, such we can hope that into this bag the food can diffuse and make the cell eating, growing, and make the tissue. As compared to bacteria, where we put the food into drop totally enclosed, now for the cell to grow tissue, they are going to grow until they hit the boundary of the capsule, so you need food to go through, as in your body. There is no blood vessel to bring the food. It's brought by diffusion as a nucleating tissue in any part of your body, whatever. It has to grow through diffusion. So a bit of movie. So we are going to do a game like this. So dripping, jetting, some by a double emulsion with a core and a shell, so we can make more complex. And it has to polymerize when it hits a calcium solution. So there is a few tricks before going to the experience of making tissue. You have to know how this hydrogel works. So they are complexing by calcium through basically it's a solubility product. It's governed by solubility products. It's a liquid-solid transition. A little bit changed by the fact there is polymer that links the site of cohesion. That's it. But it's for exactly a solubility product theory. Okay, so the question is, how do we do this? The problem is making this double structure that flows through the fluidic system with a shell, flowing down into the calcium and making polymerize without being destroyed. So this is what we developed with our colleague. And here you see that if you drip a drop into a solution, it's always mixed up with some classical vortices. And so we work out how to avoid this. And we find the solution by playing with a bit of surface science. Look at that. Just by adding a little bit of surfactant into the drop, you avoid all type of vortices. So it's like putting surfactant into the drop, which is falling into the liquid, a traces of surfactant. It's like making a protecting layer. So sometimes the surface science is quite magical. You see at the level of almost 100 milliseconds, there is absolutely no mixing. So we just try to understand that, we more or less did, and we applied it.
What's wrong with vortices? Why do you want to get rid of them?
Because if you have vortices and if you want to maintain the core shell structure to polymerize after collision with the calcium bath, it will be destroyed.
And is it important that you don't have any calcium inside the droplet?
To start, no, because polymerization will avoid any templating.
Right, I thought there's this other trick where you just put calcium in your droplet and then you drop it into an alginate solution.
This will never be possible to play in biology because the calcium will kill everything. So I'm hiding most of the detail, but we want a technology that will be able to make 1,000 capsules per second, encapsulating any type of cell that I directly grab from your body. That's what we want. That's what we are after here. Take your cells, prepare them, inject into a machine, I will show you, and get something totally clean, cells alive, happy, making tissue. So better avoid any type of chemical, any type of constraint. Okay, so since we have that trick, okay, now we do the job, we put the thin layer of alginate, we drip it into the calcium chloride solution, and you see that it's polymerizing without destroying the structure, and we are done. So this we did play a lot with that now. And for instance, I want to show you now these bags, how they reswell osmotically. That takes 10 minutes for a 2 millimeter capsule. So you see that the layer the membrane is very thin, look like plastic, but it's a little bit elastomeric. And it reswell like a balloon in about 10 minutes. So that's the time scale for the diffusion when there is osmotic mismatch. So you see the type of object. The funny thing is you do it just almost, it's done automatically. There is no effort. And it makes a nicely wrapped system like a balloon. Okay, so what do we do with this? Of course, right away we said, oh, this is magical to make a new type of plating. You can make a single cell, you end up with a monoclonal colony into a balloon, and you can have that in 3D. So you can make 1 billion of monoclonal colony out of that. So it's a way of screening and sorting and so, but I escaped this. And I just will end with a few slides about how we make this tissue and so on. So the first thing to do this job is to make the capsule much smaller. Why? Because if you want to really probe the tissue growth, you have to be at the small scale enough, 200 micron maximum, 300. Otherwise, diffusion is not fast enough, and you are essentially probing how cells get dead into a system without enough of food. So you have to be absolutely fed by diffusion with no limitation, so small enough. So this is what you can do with this type of jetting system by arranging the piezoelectrical vibration here at the right frequency given by the Rayleigh stability. So you shoot with the camera, you look the major frequency, you excite at that frequency, and you get very monodispersed drop. You have to put a field at the exit to avoid coalescence in the jet. So it's a beautiful work from Bremont and my collaborator in Paris. I'm escaping most of the movie, but the movie are really cool to look at. Okay, I'm there. Okay, now we are almost ending the talk. Now you're starting with a cell, one cell, a few cells, you put them into the bag. I'm not showing you the movie from the beginning. Now the spheroid is starting to build up into the capsule. This is a CT26 cancer cell, very known from biologics. And look what's happened into that. Okay, so the time is over there from the 0. Huh? So you see that the cells are dividing at that hour. So and then after some point, it hits the boundary of the capsule. And you see that something is happening. The dynamic is much slower in the middle, very active at the rim here. And this is a perfect object to sense the force that a tissue exerts when it grows. And this was a more or less addressed question with some other technology, but this one is without any artifact because there is a perfect control of potential, chemical potential everywhere. So we should be able to measure these stalling forces. Okay, so what do we find is the following. When it hits the boundary, you see, the dividing or the rate at which the stretch of the membrane occurs, it's much slowed down, of course. And when you burst the membrane, it keeps growing at the same rate as it was before touching the wall. What we found second is that when there is a wall, when the spheroid has to fight against the stress, there is a normal reaction of a tissue which is killing the inside and let only the rim outside alive to push under the membrane. And I will show you that. That's very, very spectacular. If you look under the movie, you see what? So there is some labeling of the cell. So everything where you don't see border has been destroyed. This is a sort of mix of debris and polymerized fibronectin. And everything which is moving is active and dividing. Okay? So this is giving you a sort of sense about the dynamic at the rim. Look at how these cells become extremely motile at the rim. And this has been reproduced, and it seems to be a really traduction of the stress they are feeling, and they are changing their phenotype to be extremely motile with a very typical lamellipodia and so on. Okay, this one I skip it, and the very last is this one. This is the one I prefer. So this last experiment has been done the following. We did a thin enough layer of hydrogel around, such we knew at the end the stalling force that it's able to produce, so we knew that it will burst it. So you will see that the membrane is going to be burst, and you will see what's going to happen here. So you see that it's growing, then it's stretching the membrane, it's an elastic polymer bursting here, so the layer is escape, and you see all the rim with these very motile cells that are invading most of the system. So it's somewhere a method to sense the force that the tissue is able to do, and there is many questions that we could address, but in all the time it's a sort of niche which is perfectly suited for a karyotic cell to create tissue. And the program about this material is absolutely huge. Just as a word to finish, we are now developing these things to create out of human body, piece of liver at a very high throughput rate. And the liver that we are reconstructing into this capsule has exactly the same phenotype as in the body, and it's directly used to make toxicity screening on drugs before going into the pipe of development and so on. So this is a big project of co-development with pharmaceutical company, Sanofi in France. And just one part of the story. I think we could go in many other directions. I suppose I should, I have other stories, but you know, I prefer these two ones who were nicely understood than talking in the emptiness. Thank you very much.
Questions?
Yeah.
I had two questions about the very last thing you showed. One is, if you start with the cells, but they were never encapsulated, they don't display this motile behavior.
No.
And my second question was, what makes you confident that this is a phenotypic change and not just like a behavioral change in some way?
That's the same, if you want. When I talk about a cell, if I see the behavior changing, I have to tell that the phenotype has changed.
So, to pull another biological example, if you took Dicty and you started them and they formed this collective slug, you're familiar with like the slime mold slugs?
No.
No? Okay. But just like if it's the same cell and it just becomes more motile, that counts as a phenotypic change?
Yeah, it has to. And there is also testing on this cell that suspect that it is. Yeah. Still a lot to do, but good chance that it is.
Is the material of the hydrogens? That's the component of the hydrogen.
Sorry?
The component of the hydrogen.
I don't hear
The
you.
molecule of the hydrogen.
The algae.
The hydrogen, yes.
She wanted to know what the capsule's made of.
Ah, it's a alginate, yeah. It's a bio-grade alginate.
The alginate could be like the feeding molecule for the cells. This is the reason why the cells are moving. Because
Yeah, okay, but if you look at the cell before they feel the membrane and feel the pressure, they don't have at all the type of.
Because generally, for example, for bacteria, if you want to make sense the bacteria to the external environment, one of the most easy stimulus is like giving them a little bit of sugar, so they can move because they are attracted.
Yeah, but there's the food that we give to the cells, to this. They are human cells, so there is far more than sugar. So what is in the alginate is, first of all, even bacteria don't eat alginate. That is the great stuff about alginate. Some microorganisms do, but most of them don't. Yes?
Let me repeat the question that was posed before. I mean, suppose I have one and the same cell given two different inputs. It will react differently to input A and to input B because these are different inputs. Why do you interpret that as the cell changing its phenotype? I think that was the question being asked. So,
Yeah, so you're talking about the last part again, huh?
No, but I think the answer, which I think you started saying before, is you give the same stimulus and you get two different responses. Those are different phenotypes.
that I understand. But you, in response to the question there, you seem to say that if you're given two different inputs, you interpret that as two phenotypes. Maybe it was just a misunderstanding of the question.
No, that was my question, basically.
It was your question?
Because one cell
Yes.
never feels the stress of the wall, and then once it feels the stress of the wall, it becomes motile. And so I was confused as to why going from non-motile to motile.
Okay, so I will try to explain a bit better. When the spheroid grow without pressure, everything is the same, no worries. Once you start to hit the wall, then it experiences a pressure.
Right.
Okay. This pressure is changing the whole system. Two consequences. First, the middle is turning into necrosis, and that's repeated, and people understand that. And the rim is turning into a new phenotype, extremely motile. That there is two consequences.
But how, it's still,
After the thing bursts, the children are more motile, right? So it's not a memory effect, right? Because that's a new cell. So something switched about the gene regulation.
so because it's a permanent change and not a change that is linked to the current stimuli.
No. So, but about all that, there is still a lot to deep and to understand. It's just a phenomenological result that we did repeat. So far, that's it.
Oh, wait. I understand now. Yeah, I missed the part where this was heroin. Okay. Yeah, exactly. Where?
Other questions? Okay, let's thank